I think I saw one more participant joining just there, but I think looks to me like the numbers are leveling off. So I'm going to start us off. So first of all, welcome to everybody to this, which is a Global Alliance for Genomics and Health Fireside Chat. My name is Susan Fairley. I'm the Global Alliance for Genomics and Health's Chief Standards Officer. I'm based at Embley BI on the Genome campus just outside Cambridge in the UK. And today it is our great pleasure to be joined by Chris Wigley, who's the Chief Executive Officer of Genomics England. And he's joining us for the second one in this series of virtual fireside chats. The format originated, I believe, at our face-to-face meetings, and it kind of gives an opportunity to listen in a kind of slightly more expanded format to the thinking from somebody who's a leader in genomics, and also gives an opportunity to ask some questions. We were really fortunate to start off this series last year with Catalina Correa Lopez. And as I say, it's my pleasure to have Chris joining us today for this one. The format is a kind of semi-structured conversation. There's a number of topics that we're hoping that we're going to get through. We're hoping that Chris is going to be able to tell us a bit about the Genomics England story so far, also about some of the really exciting current projects that they're involved in, and also kind of looking more to the future about some of the challenges associated in bringing genomics into health, and then also a little bit more about kind of more specifically some of the challenges on the research side. Questions are very welcome. I think you should have a number of options for doing this. I think there's both a Q&A facility and you may also be able to use chat, but we'll do our best to keep an eye out for questions. If you do have a question for Chris, please feel free to let us know. And other than that, just to say that we will aim to close within the hour. So other than that, I think that takes care of all of the housekeeping. Again, Chris, thank you very much indeed for joining us. It is our pleasure to have you here today. And if it's okay with you, then maybe we can make a start.
Yeah, thanks, Susan. It's great to be here.
Okay, thank you. So as I said, one of the things that we're hoping to talk about is a bit about kind of the story of Genomics England so far. It's clearly a leader in bringing genomics into healthcare, but people, particularly maybe those outside of the UK, may not be familiar with its origin. I was wondering if you could tell us a little bit about that.
Yeah, absolutely. Great place to start. So we obviously do a lot of science and technology work at Genomics England, but we talk a lot about human stories, about the reason for us coming to work, the reason for us doing the work that we do is to try and impact as many human stories as we can, and to effectively try and help as many people as we can. And actually the origin story of Genomics England is really an interesting human story as well. So if we cast our minds back to the sort of early noughties Human Genome Project, you know, hundreds, thousands of scientists, not least based in the campus just outside of Cambridge, and worldwide effort, you know, multiple years, multiple billions of dollars to sequence that first one genome. If we go forward a bit, we start to sequence a few more, but by probably about a decade after that, which is when Genomics England existed, the numbers of whole genomes out there was still relatively modest. So there are no slides. Sorry, I'm just seeing in the chat, is the camera off? Can't see slides. But hopefully you should be able to see Susan and I. Yeah, so if we think then to probably about a decade after the human genome being sequenced, there's still relatively modest cohorts of whole genomes out there. As part of the Olympic legacy, there was discussion around, could we do something in this space? At the same time as that happening, and back to sort of human stories, the Prime Minister of Great Britain at the time was David Cameron, and the Cameron family had a kid, Ivan, who had a rare disease, Ohtahara syndrome, which is genetically driven and which was not fully understood. And like many families, like many parents, the Camerons got pretty smart on what was happening with their kid, with the science around that and so on. And the scientific leadership of the UK at the time, particularly John Bell and Sally Davies. So John Bell, you know, Regis Professor of Medicine at Oxford. Sally Davies was the then Chief Medical Officer. Discussed with Cameron and said, look, one of the big challenges we have in bringing the kind of insights from genomics into a way that that can be helpful for people both in research and in healthcare is that we need more data. We need to understand what normal looks like in a genome, what unusual looks like, and it's only by generating that kind of that truth set of data that we can start to generate meaningful insights for an individual human being. And that really was the very early origin of the 100,000 Genomes project. That was announced as part of the Olympic legacy, and Genomics England was created as a government-owned company in order to deliver on that project, the 100,000 Genomes project.
And the 100,000 Genomes project, I mean, there was a publication on the rare disease pilot in the New England Journal of Medicine at the back end of last year. Could you tell us, I mean, it'd be really interesting to hear more about that project, and also about, kind of how patients have responded to it and the impact that it's having.
Absolutely. So the way it was set up was that this was to try and generate insights for the most complicated cases out there, people with hard to diagnose rare diseases, people with cancer. What could we learn from engaging with them, sequencing their genome, linking that to their clinical data that could help those cases? And Ewan Ashley, who's a professor in Stanford and does work along these lines, describes it as like the Sherlock Holmes cases. Where can we find all of the clues that will help us to understand what's going on with someone and how we can best help them. So around 3/4 of the participants in the 100,000 Genomes project had undiagnosed rare diseases. Around between a quarter and a third had cancer. Particularly for those with rare diseases, they had often gone through what we call the Diagnostic Odyssey. So lots of different appointments with lots of different consultants, symptoms that may be having a real impact on their life, but can't yet be sort of linked to a specific condition or a specific treatment program. And that could often be multiple years. I mean, some of the participants who came into the 100,000 Genomes project had in one case over 100 consultant appointments for this undiagnosed rare disease, often dozens or scores of appointments, often multiple years. And the idea was really to say, with this one molecular test of sequencing someone's whole genome, could we shortcut that diagnostic odyssey and actually get to diagnoses? So it was really important to obviously engage with patients and explain the concept and bring them in. As part of that, it was a big part of the success of the 100,000 Genomes project was really the partnership with the NHS. So engaging with those clinical communities, often clinical academics who were both treating patients but also doing research themselves. And that, I think, is at the heart of what Genomics England is trying to do, is act as a bridge between the world of research and the world of kind of healthcare delivery. And so from the way that participants were recruited into the 100,000, all the way through to the way that we work today, that linking of research and healthcare is at the backbone of it. And for the patients, in terms of how patients responded, it was hugely powerful for them, for their families to often get to diagnoses. So around, depending on the condition, between sort of 10-20% up to sort of 40 or 50% of participants got to primary diagnoses. And that is hugely meaningful at a human level. Even ideally, you get the diagnosis and then that leads to a treatment pathway, and actually we can get to a better outcome. And that's where we want to get to ultimately for everyone who's a participant in these kind of programs. Even just getting a diagnosis though is so transformative for an individual or a family, because as a friend of mine would put it, you know who the enemy is now. You know what's going on. It may well be that even if there is not a cure, so to speak, there are different interventions we can make that will improve someone's quality of life. There are often communities out there that you can join up with, energize around particular conditions. Once you know you have something, you can try and lobby for or kind of marshal resources for research into your condition. And so all of those things can be really meaningful. And at a very human level, I was really struck. The mother of one of the participants, Owen, when they got the diagnosis for Owen, who was a kid who'd had a range of kind of developmental problems. That was so meaningful for her, because she said she'd been carrying this kind of burden of guilt of like, was it something that I did? You know, was it my fault that Owen had had these problems? And we're all so kind of crazy as human beings. You know, we can tie ourselves in knots about these things. Actually getting to that diagnosis, we know what it is. It was a de novo mutation, just like a 1 in a million shot. And that sense of relief is maybe the wrong word, but the release from that sense of guilt that it was something they had done that had caused these problems for Owen was huge. So that you know, that work is ongoing. One of the legacies of the 100,000 Genomes project was the way that we've engaged with participants, and we can maybe touch on this a bit later as we get into things like data privacy and ethics and so on. We're continuing to work with research groups who continue to look at the undiagnosed conditions and cases. So that work will continue. But in many ways, that sort of laid the foundation for all of the work that we're doing today.
And I guess also that kind of leads nicely on to kind of the next thing, because obviously Genomics England, as you've said, the kind of reason for it coming into being was pursuing the 100,000 Genomes project. But we can now see that Genomics England has moved to now supporting many different projects. I wonder if you could tell us a little bit about how you've been able to make that transition and use those foundations from the 100,000 Genomes project to now support in a number of different projects.
Yeah, absolutely. So we talked loosely about this process of transitioning from being a project organization to a platform organization. And that really involves us trying to enable the whole genomics ecosystem in the UK by doing, I guess, two big things. Big thing number one is working with the genomic medicine service in the NHS, doing whole genome sequencing for diagnostic purposes for people with rare disease, people with cancer. And so that's live and running now. And that generates lots of better patient outcomes. You know, that's its principal purpose. It also generates lots of data. We can then de-identify that data, make it available to researchers in the trusted research environment context. They can do their work, whether that's on kind of fundamental biology, whether that's on new diagnostics, new therapeutics, working with biotech and pharma companies as well. And then we can feed those insights back into healthcare, whether that's at the level of an individual diagnosis, whether that's at the level of new treatments, new pathways, and so on. So those two big services, effectively providing a service to the NHS and a service to the research community, act as the foundation for everything that we do now. It's fair to say that there were a number of there was a lot of change involved in making that transition from project to platform, at the level of you know, how we'd set up the technology, how we organized ourselves as an organization, the kind of timelines that we were working to for sequencing, interpretation, analytics, and results, and so on. So there's been a lot of change over the last few years. We've grown quite a lot as an organization. We're now about 400-ish people, and we're, you know, running live services in the NHS with kind of 99 point whatever availability, in a way that at the beginning of the 100,000 Genomes project would have seemed, you know, almost unimaginable. the pipelines, the sequencing operations, the interpretation really are live services with hundreds, thousands of users, and that's been quite a big journey.
Yeah. And it sounds an amazing journey. And I guess to kind of come back on one of the things that you touched on there was your work with the Genomic Medicine Service, who I think you're working with people on cancer and rare disease. I mean, I was wondering if you could tell us a little bit more about what that's been like and again, the impact that you're seeing that work having.
For sure. So we launched into live clinical testing of the service over a year ago. So this was November 2020. And we've had thousands of patients come through the service. So there are a number of indications or kind of symptoms that if someone is presenting with those symptoms and they don't have a diagnosis, then they are now eligible for whole genome sequencing kind of on the NHS, which is great. And that's often in areas like developmental disorders, musculoskeletal conditions, sight conditions, neurological ones, neurodegenerative, whole range of conditions where people can be now eligible for whole genome sequencing. And in cancer, any kid or young person in the country, which is England, so this is a partnership with NHS England, although we also do work with the devolved authorities. Any kid or young person is eligible for whole genome sequencing as part of their standard of care for cancer. And also any blood cancers, so hemonc, neurology, sarcoma. We're looking at things like triple negative breast, ovarian going live. So the number of conditions is growing as we kind of demonstrate the value of doing this. And having this now as a live service in the NHS as part of routine care is huge. So it's the first time that any healthcare system in the world has offered this as part of standard of care. There are some of the fancy private hospitals in the States where this is absolutely part of the care package, but you need amazing health insurance or an amazing other source of private wealth or whatever to tap into that level of treatment. Whereas here, in line with the founding principles of the NHS, free at point of care, equitable access to anyone in the country, which is amazing. And in fact, I had an email from a cancer an oncology consultant a few weeks ago who was talking about the impact on one of his patients, well, the family of one of his patients. So this was a family with 6 kids, not a well-off family at all, and one of the kids had developed cancer. Back to the Camerons getting smart about genomics, the mum had researched this a lot, had understood that for the type of cancer that her kid had, whole genome sequencing was going to give the best insights possible. And being a sort of organized and dynamic parent, had turned up to the meeting saying, right, I know that the best thing is going to be whole genome sequencing. I know that's still really expensive. I'm starting a crowdfunding effort. We don't have the money to pay for this personally, but I'm going to do a charity run and I'm going to raise the money. And if we raise the money, can you do this for my child? And he said, look, you don't need to raise the money. It's on the NHS now. It's this is part of the NHS's treatment for kids with cancer. And he said that she broke down in tears, was amazingly, the sense of relief of this is now something that is part of routine care in the NHS was just extraordinary. And that then having done the whole genome sequencing, that then did inform the treatment of that kid to a much better outcome, which is huge. So it's hearing those kind of stories of really help bring to life the benefit that these incredible advances in science and technology and clinical practice and so on. If we can focus all of those advances down into that conversation between a patient and a doctor, it's so powerful. And then if we think that family is one family, but actually any family with a kid with cancer now in the country is eligible for these.
It's incredible hearing about the impact it's having on routine care. I suppose something else that's coming to mind as you're talking about, hearing stories about the impacts of genomics, something which was not a few years ago routine, obviously, was COVID. And COVID has become, just this huge thing. Genomics England has had involvement in the UK's response. I was wondering if you could tell us a little bit about that.
Yeah, for sure. It's interesting. When Genomics England was founded, it was initially framed as kind of 3 areas of investigation, which were rare disease, cancer, and infectious diseases. The way that patients ended up being recruited into the program meant that particularly through the 100,000, there was much more of a focus on rare diseases and cancer. The COVID pandemic appeared, and actually suddenly that sort of third leg, so to speak, really roared into life around how can genomics help us with insights about how to manage the pandemic. There's obviously two big and distinct programs of genomic sequencing. Huge program around GA4GH, which the Sanger is hugely proud of the role that the Sanger has played in that, around viral sequencing, spotting the variants and so on. We were looking at human sequencing and what could we learn from someone's genome about their propensity to get COVID really severely. So in the first few months of the pandemic, we would see people who, quote unquote, looked in many ways the same, whether that's in terms of age, ethnicity, other comorbidities, other characteristics. One person would be absolutely fine, would feel like they had a cold. Another person would end up in intensive care, would be really, really sick. And so the study design for the work that we did in partnership with Kenny Bailey at the University of Edinburgh, with Illumina, with LifeArc, with many other partners, was focused on recruiting a cohort of people from intensive care units, so who had COVID badly enough to be in intensive care. And so huge shout out at this point to the clinical research nurse community, who in the middle of the nightmarish scenario of the first wave of COVID were, as well as doing everything else they were doing, were recruiting and consenting patients onto their study, which is extraordinary. And then, so that's kind of one part of the study. The other part of the study is volunteers who've had COVID but been asymptomatic or had it only really very mildly. And then doing the kind of compare and contrast, are there biomarkers that are associated really clearly with severity of response that we can pick out by comparing those two data sets. And so the work started recruiting really rapidly. We've done kind of multiple rounds of insights from that. The first, I think overall now it's over 30 new locations on the genome that are associated with severity. The first wave of insights led to the identification of four targets on the genome that were already seen as druggable. So that suggested candidates for the recovery trials. So that could be interesting candidates for repurposing for treating people with COVID, and three that were not yet druggable. And so that link up of and that fed through straight to clinical care, which is amazing. But I think that the link between the work that GA4GH were doing on the viral genome sequencing, we were doing on the human genome sequencing, React from Imperial were doing on the kind of broad spectrum population screening, ISARIC were doing on the sort of clinical care, the recovery trial people were doing on the rapid basket trials of different approaches for COVID. That really was kind of the ecosystem at its best, rapidly generating insights, rapidly turning those into clinical practice, rapidly having impact on patients. I mean, dexamethasone being the sort of standout example of that. And I think part of what that's inspired us to do at Genomics England is to think, how can we as an ecosystem kind of sustain that level of focus and energy and drive that we had, and collaboration, and a really clear sense of mission that we had through the pandemic, and bring that to the sort of sustained development of insights and approaches that will help improve people's wellness and health, going forward.
It's really incredible, and I guess also kind of looking to the forward-looking stuff. You've also got, as what we've already discussed, a fairly substantial quantity of current work, but there's also a whole set of new projects that Genomics England are getting underway. There's, I believe, your Cancer 2.0 initiative, which is making use of the really exciting developments we're seeing in long read technologies, work with newborns, and of course the very topical diverse data project. And I was wondering if you could tell us a little bit about what's led to these projects, and maybe a little bit about them.
Yeah, so I'm smiling because I was talking to one of our product managers recently, and she was updating me on all the progress that they'd been making. And I said, look, is there anything I can do to be helpful with all of the great stuff that you're doing? And she said, yeah, you know what, there is. Can you just not have any more bright ideas about other stuff that we can do? No, which is good feedback, which I took on the chin. We're incredibly excited to be building on top of those two kind of core services, the genomic medicine service and the research work with these pieces. In the recent spending review round, we've now got 3 years of funding for them, which is amazing. And the Newborns and Diverse Data projects headlined Rishi Sunak's announcement of the $5 billion healthcare R&D fund for this spending review, which was really exciting, particularly because I think we were not sure that people would get the vision around newborns. It can feel like an area with tons of ethical mines or minefields that we need to navigate really carefully, which it is. And at its most expansive, it's kind of a 100-year vision. But so the fact that people felt like this was something to get behind was really exciting. So in fact, maybe if I talk to that first and then can touch on a couple of the others. So with the newborns piece, the origin of this really goes back a number of years, again to Dame Sally Davies when she was CMO. She asked the question of Genomics England, would it be interesting and useful to do a whole genome sequence alongside the sort of heel prick test that babies get when they're born, which tests for 9 things? Could we use a whole genome to test for hundreds of things at the point of birth? It's particularly highly penetrant conditions where if you have the variant in your genome, you will have the condition that affect kids early in life, and where if we can treat early, we get to much better outcomes. So that there's already been quite a lot of work in a low-level kind of way on the science and on public dialogue around this topic. How do the public feel about this? Are they supportive? What kind of concerns do people have? How can we work with the public to design this kind of program? What we're now doing with the launch of this as a major program and with the funding and so on is kind of 3 big work streams. The first one is this screening at birth piece. So alongside the heel prick test, we know that there are 200 to 300 conditions that are highly penetrant, that are treatable, and that affect kids in early life. And the range is driven by things like, do you take a cutoff at 5 or do you take a cutoff at 8 and so on. So we're working through what the final list will be. It's kind of 200 or 300 conditions that that we can get ahead of by spotting them at birth, getting the kid straight onto a treatment plan. And so that's kind of thing 1. Let's cure these sick kids. Thing 2 is, we know from the science that there will be lots of kids who are born with potentially pathogenic variants or known pathogenic variants, but for conditions for which there isn't a treatment today. So as we were talking about earlier, getting to a diagnosis is great. That can be really helpful, and it's much better than not having a diagnosis. But the next step is then, okay, so what do we do about it? So the second work stream here is about 4 highly penetrant conditions that affect kids in early life. Can we work with biotech and pharma on a sort of therapeutic innovation platform? So coming up with new treatments, new drugs, new therapeutics, new interventions for conditions that are highly penetrant, but for which we don't currently have a treatment. And particularly interesting and exciting work right at the cutting edge of science around gene therapies, and also around kind of small n or even n of 1 drugs for these kind of rare conditions. So that's kind of thing 2, is research into new treatments for those conditions. And thing 3 is the most I guess sort of nascent or experimental part of this, which is what does it look like to have a lifelong relationship with the genome? So even if you're a kid who's born perfectly healthy, the cohort is 100,000 kids. 100,000 seems to be a number that we like at Genomics England. And this will be the first cohort of people in human history who are born with their genome on file. What does that mean? How can we use that to help understand wellness for those kids as those kids, if they rock up in intensive care in early childhood, rather than having to have a conversation about sequencing, have a parental parent choice, get a blood sample, extract the DNA, sequence the DNA, get the insights, feed those insights back to a doctor, blah blah blah, all of which, even with a fair wind, takes weeks. Their genome is on file there, and it's immediately accessible to a medical team who's treating them if they're in intensive care as a child. Into teenage years, there are really interesting questions around mental health, around obesity, around things like hereditary diabetes. Could we get ahead of some of those conditions and help people have the best outcomes they could have? Into middle age, cancer, cardiovascular disease, general wellness, later years, kind of neurodegenerative and so on. So at its most expansive, it's kind of a 100-year project to try and understand both what kind of medical benefit or insights can we derive over the course of someone's life from having the genome on file, but also as a society, like, what questions do we want to ask? What questions do we not want to ask? What things do we want to explore? What things do we not want to explore? A good example of a grey area where we don't have a view, but it's an interesting question to explore, is things like educational attainment. There are some people who talk, I think, often quite blithely about things like genius spotting. We should sequence everyone at birth so we can spot the geniuses and kind of, support the future Leonardo da Vinci. It's like, yeah, maybe, I don't know. But from projects like Deciphering Developmental Disorders, the DDD project from 100,000 Genomes Project, which had lots of kids, we know that there are certain variants that are associated with developmental disorders or intellectual disabilities. And those reach a clinical threshold that say, right, this is someone who in clinical terms has a condition. What if someone has, a subset of those variants, right? What if they have something which doesn't reach a clinical threshold, but will meaningfully impact how they learn? What kind of style of learning they need, whether they need extra support to have the best educational outcomes. Is that an interesting question as a society that we want to explore? Would we, in one world, screen kids to say, how can we optimize your education? Don't know. It may well be that people are like, oh no, gross. Like, no, thank you.
It's interesting as well, because I think this is a question that we're seeing coming up elsewhere. I know certainly in GA4GH, I think our regulatory and ethics work stream, this is one of the questions or one of the areas that they're really interested in at the moment as well. Yeah, and I guess also just briefly mentioned the two other new projects, Cancer 2.0 and Diverse Data.
Yes, so I know we could probably talk for like 5 hours just about each of these, so I'll try and be briefer. But so the Cancer 2.0 initiative is really about how can we bring to bear like the latest technologies and kind of emerging treatments to optimize cancer treatment for people who are in the flow. So you mentioned long read and methylation, long read sequencing and methylation insights. So we're working with Oxford Nanopore Technologies at the moment on, I guess, effectively like validating and kind of seeing what insights we can derive from doing that in parallel to the existing standard of care cancer treatments. We're also looking at things like liquid biopsy, and we're exploring with NHS England doing a kind of bake-off of all of the different liquid biopsy approaches. So that kind of clinical validation of new technologies is kind of work stream 1 within cancer, and then work stream 2 is more on the research side, which is multimodal data. So in partnership with the National Pathology Imaging Cooperative, I always get the C wrong, I always think it's centre, but it's actually cooperative, in Leeds, who have hundreds of thousands of cancer images and who have very kindly scanned all of the radiology and pathology images for the 100,000 Genomes Project cancer participants. The goal there is to say, right, we have the genomic data. We now have the imaging data from radiology and pathology. We have the clinical data. We have the really richly annotated cancer registry data. Can we start deriving features from each of those sources of data to build better predictive models around what's the best treatment option, what's the best clinical trial to get someone onto? Can we do lookalike modeling to say what do patients who look like this across various dimensions, what happened to them, what treatments did they go on to, what would that suggest about the optimal course to take for this patient? So very much in the research realm at the moment, but as ever, if we can kind of harden some of those insights from research and bring them into clinic, that's the ultimate goal. Finally, on diverse data, there's this kind of long-standing issue in the genomics world around representation in the large genomic data sets that a lot of the studies are using to derive insights. And there's been a real pickup in activity and discussion around this recently, which is great to see, and we've been trying to stimulate that debate as much as we can. One of the statistics that sticks in my head is I think it's like 85% of GWASs that are out there in the sort of genome-wide association study kind of catalog. 85% of those also are based on cohorts of European ancestry folks.
Yeah, I mean, there's the work by Alice Popejoy and others, which lay that out. Yeah,
completely. And so just the analogy that I often draw is to things like machine vision algorithms, for facial recognition and so on. When those were starting to be developed using AI a few years ago, there were all these issues of, because they've been trained on data sets of white male faces, they were much less good at recognizing women, they were much less good at recognizing people of color and so on. And that was really bad, but to some extent, okay, it's a facial recognition algorithm, like maybe the facial recognition algorithm doesn't work so much or whatever. If we're now talking about, we're going into an era of personalized medicine, where actually your life or your quality of life could really be determined by, are we going to get you onto the right treatment for you? And we're using genomics as part of the mix to get to that best answer. If those genomic insights are not as relevant for people who are not from European backgrounds, that's like a massive issue. Our mission statement is around helping to create a world where everyone benefits from genomic healthcare, and that word "everyone" is really important. So for us, we were really excited to get the funding to pursue this at scale. The Secretary of State, Sajid Javid, has volunteered to be participant number 1 in this cohort, which is great. And the goal over the next few years is to try and make sure that we get to kind of statistically powered cohorts for each of the major ethnic groups in the UK. And that's hopefully also incredibly relevant for research globally. But obviously, as a British government company, our focus is initially on making sure that the data sets that we curate and are the sort of custodians of in the National Genomic Reference Library are as diverse as the communities that we serve. So yeah, hugely exciting work going on there.
And really important for laying the foundation for genomics going into health in future, which takes me nicely through to the next thing that I was wanting to ask you about, which is, what do you see as being the major challenges facing genomics in healthcare, not just necessarily for Genomics England, but for the field as a whole?
Yeah, it's a really interesting question, and there are some kind of intellectually complex questions or challenges that we need to solve, and there are some intellectually simple but operationally complex challenges. And so I think on the intellectually complex side, there are still a bunch of questions around, which kinds of conditions, what kinds of indications and so on are going to benefit from what types of testing. And so we've talked quite a lot about whole genome sequencing. If we think about someone who presents to the National Health Service with cancer, let's say, there are single gene tests we can do, there are panels we can do, there's now a TSO 500 panel with 500 genes in it, there's a whole genome sequence. You can then go beyond a classic short read 30x or 100x whole genome to long read sequencing, PacBio, ONT. to RNA, to proteomics. There's this sort of almost bewildering array of techniques that we could throw at a problem in theory, but which one is actually going to give us insights? Which ones are not? I slightly flippantly use the analogy of like the Harry Potter sorting hat. We need to get to a point where, for a patient with a given condition, we have the sorting hat that can say, right, for this patient with this condition, we should do RNA-seq and long-read methylation. For this one, we're not going to get. We should do the 500 gene TSO 500 panel, and that will get us most rapidly and most easily to the best insights and so on. So I think, particularly in the context of the NHS, a lot of the intellectual challenge is about which tests, which treatments for which conditions, at what speed, at what price, for which patients, for which conditions. So lots to work through there. A lot of the operational challenges are almost kind of more complex though than the rocket science challenges, because to some extent with rocket science you can kind of train and hire rocket scientists, kind of point them in the right direction. They do rocket science and it works, and we can get to answers. The NHS is the world's third largest employer after the Chinese army and the Indian railway system. It's an incredibly complex ecosystem of organizations, regions, sub-regions, all overlapping, all on a range of everything from like blisteringly contemporary technology to bits of paper, and it's a system which is on a massive strain, and which has only increased obviously through the pandemic, but was already under strain before that. And for a busy doctor, us, which is like a collective us of people in the NHS, people in academia, Genomics England, whatever, rocking up and saying, great, whole genome sequencing, RNA-seq, proteomics, just like what? And so the question is, how can we start to validate and bring into clinical practice these technologies in a way which they can fit into people's workflows? If we need people to get tissue biopsies in a different way, how can we make sure they have the right tools? Whether that's a freezer rather than a formalin fix for a tissue sample for a whole genome, or whatever it is, and how can we engage with, listen to, meet the needs of those clinical communities? Those are just really complex kind of operational challenges that we're working through, I guess, as a community.
And I guess also on top of that, we said it's a fast-moving space, and you were talking about the need for something equivalent to a sorting hat. I guess also working with the wider clinical community to enable them to be aware of and stay up to date with things. Because you mentioned when we were talking about some of the testing earlier on, the range of conditions for which you're able to provide support is increasing all the time. I mean, how is engagement currently working? I mean, what are your hopes there?
So there's a bunch of organizations that are doing great work in this space. So the Association of Medical Royal Colleges is brilliant. We talk a lot to Helen Stokes-Lampard and the folks there. And they're a great kind of trusted channel to different clinical communities. Health Education England's Genomics education program, which Kate Tatton-Brown now leads, is also fantastic. Really engaging materials, really great relationships with those communities. So there's a lot of activity in that space, and we as Genomics England don't want to add more complexity into that. So we try to work in partnership with those organizations to get those messages out there, do that listening, and try and tailor what we're doing to, I guess, to reduce friction as much as possible in terms of introducing new stuff into what's already a complex system. Yeah, for sure.
And I guess also, and we're heading slowly towards the end there. We've got about another 10 minutes left, but just to the kind of final few stages, to switch the focus to some of the challenges. And we've spoken about challenges in terms of bringing genomics into the healthcare space. in terms of the research side of things, what do you see as being the challenges there?
Yeah, so I mean, lots of interesting, I guess, challenges and opportunities are always sort of like the flip side of each other, right? So huge opportunities in healthcare and in research, and then lots to do to kind of realize those. I think one piece is one massive piece that we run straight into on the research side is how do we use this data that we have? Whose data is it anyway? What's an appropriate use of it? What's not? And how can we maximize the research yield, so to speak, while protecting people's privacy who've donated their data to science, and making sure that the research that's happening is relevant to those people. So the first, I think, easy to say and difficult to do, or difficult to kind of put into practice piece is, it's not our data, it's the data of the people who've wanted to give it to us, right? Which, as I say, it sounds like, well, yeah, duh, obviously, but as a broad community, let's say science, historically, we haven't always necessarily respected that, and there are various historical events, things that have happened that mean that various communities or various people are naturally quite suspicious of this. So I think the onus is on us to be kind of demonstrably trustworthy in terms of how we think about how we use this data, who we partner with, what the focus is. One of the great things that Genomics England and the participants in the 100,000 Genomes project kind of co-developed through the 100,000 Genomes project was this system of participant representation. So there is a participant panel of 30 people who are nominated by the participants in the program, and who kind of speak on their behalf. They sit on the Access Review Committee for access to those data sets. And if an academic group or a biotech company or a pharma company want to get access to those data sets in the research environment, the participants have to say yes. So if they have a veto, and if they say no, I can't overturn them. The board of Genomics England can't overturn them. It would have to go up to the Secretary of State to actually overturn the veto of the representatives of the participants on the group. So that, I think, is not just about patient engagement or participant engagement, it's about power. A hard veto like that is actual real power. And so I think the more that we can empower patients, participants in this space, the more solid ground we're on ethically in terms of what's happening with that data. Because of those systems and the sort of the ethical thought leadership that the participants in Genomics England have shown over the last few years, we've worked really happily with dozens of biotech and pharma companies who have access. That has never been controversial, because it's being done in the right way, kind of demonstrably in the right way, with the right checks and balances, with the participants in charge. So I think that's a huge piece, and we need to have innovation and trust need to go hand in hand. And actually, it's often framed as a kind of tug of war. Well, we want more innovation, so we need more latitude to do stuff. Or, well, we want more trust, so we need to restrict what people can do. Actually, I think when we get this right, innovation, like trust enables innovation, trust accelerates innovation, because it means we can confidently move forward and do things, rather than kind of second guessing what we're doing and so on. So Genomics England was a kind of early adopter in the trusted research environment model, where the researcher comes to the data. And we talk about the kind of aquarium model where you can admire the fish, but you can't take the fish away with you. And that's a big change. So I mean, lots of people at EBI, for example, will be used to working on an on-premise environment with an HPC capability that they can book to kind of run their jobs for them and so on, but working on data locally. I think there's a multi-year community kind of engagement challenge that, for example, UK Biobank are right in the middle of now as well, as they've adopted the trusted research environment model. How do we create trusted research environments which create a good working experience for researchers? And how do we kind of co-design those with the research community, and how do we bring people on a journey from kind of existing established ways of working to kind of new ways of working, which have all sorts of benefit in terms of scale, in terms of the ability to federate across different data sets, in terms of the ability to do these things on massive data sets in a way which is demonstrably trustworthy and so on. But it's a big change for the community.
Yeah, and I guess it also kind of leads us through to kind of the ideas around about data federation. And I suppose this is the point where I put my chief standards officer hat on and start to think about the roles of standards in engineering. Because Genomics England has obviously been really kind of leading the way in many respects in terms of bringing genomics into health, but there's also this business of kind of working with the broader community to support federation as well.
Yeah, and I mean so I think the good news is that everyone, when I say everyone, I mean things like people with budgets and like engineering teams are
it tends to help.
Getting on board with this, which is great. It hasn't quite been finalized yet, but I think there is going to be some substantial funding in this spending review for federation across like big research TREs in the UK, and so EBI, GEL, UK Biobank, over time, Our Future Health as that comes on stream, NHS Digital. If we can start to make sure that we don't end up in a world of just a bunch of massive data silos, that's going to be transformative for research. If we do end up, as more and more organizations adopt the trusted research environment model, if we don't solve the federation problem, we're going to be in a really painful position that's going to hold research up. I think that's a really interesting question that we probably shouldn't get into now because it'll take too long, but like, what is the right number of trusted research environments in the UK? I think there is a world in which we suddenly have hundreds, which I think is mad. I don't think we can ever get to a world where there's just one. But I think the answer is probably somewhere between, I don't know, 10 and 15, depending on use cases or something. But there are definitely people who think that, for example, every hospital trust should have its own trusted research environment, which I think is hugely challenging. They should obviously have the ability to do research, the ability to access trusted research environments. But I think a lot of the challenges we're facing now in terms of implementing genomics in healthcare, which we were talking about before, are down to choices that were made about technology architecture 15 years ago. Let's not make choices now that mean that in 10 or 15 years time, people are cursing our names and saying, why do we do this? So as you say, I mean, we're hugely supportive of this agenda and of finding the right way to adopt both like common standards, common architectural decisions, common data ontologies, and it's really tough. I mean, it's really difficult. All it takes is someone using a slightly different ontology for phenotypes, and suddenly it's just tons and tons of manual labor to try and rebadge stuff from one taxonomy to another taxonomy and so on. But I think the more that we can do around validating a researcher, so I really like this analogy of the passport and the visa, right? The passport validates that this researcher is who they say they are. They're bona fide, person from Oxford, Harvard, Warwick, wherever. And then you have a visa for the Genomics England environment, the UK Biobank environment, the ONS environment, wherever. And we start to build those bridges in terms of common ontologies, ability to do meta-analysis across multiple data sets and so on. I think we have to be doing that in parallel to the move to trusted research environments. Otherwise, we end up in a really painful and sort of fragmented world.
Well, I'm noticing the time. Really sorry to see that we are approaching the top of the hour. I have certainly learned lots about Genomics England. I've thoroughly enjoyed it. I haven't seen any questions from attendees. So this is the final opportunity if you do have a question, but I'm guessing that if anybody had questions, I think you've already done a wonderful job of telling us lots more than we knew at the beginning of the discussion about Genomics England. So I think this may actually be one of those rare cases when it's been a pleasure to listen to you. Again, thank you very much indeed for joining us and for giving us your time. it really has been fascinating. And yeah, just thank you once again. And
No problem.
I'm sure that the rest of it, yeah, and I'm now seeing a number of messages of thanks coming in on chat. So clearly
and
that's been
I'm on Twitter as Chris_Wigley, and my DMs are open if anyone wants to follow up. So feel free.
well, thank you very much indeed.
Thanks all.
Okay, thanks everybody. I think we'll close things there, but thank you very much indeed. Bye everyone. And once again, thank you very much, Chris. Bye-bye.